Hematopoiesis in bone marrow can be studied either in histological sections or in smears. The earlier image I showed you of the blood sinusoid was from a histological section through the marrow in the vertebral body. Bone marrow smears, made in a way similar to blood smears, allow a greater degree of differential staining than is possible with histological sections. Also, the cells are flattened and have larger diameters, so smears are more useful in clinical diagnostic purposes. On the other hand, histological sections preserve the histological structure of bone marrow. Firstly, I'm showing you a bone marrow smear in this image. The slide was prepared by smearing a drop of human marrow tissue across a glass microscope slide. The cells adhering to the surface were dried and stained with Giesmer stain, a special stain for bone marrow. As with hematoxylin and eosin, acidophilic structures stain red and basophilic structures blue. Inevitably, a substantial amount of blood is collected along with the marrow, so it was important for me to avoid these areas of the section here that I know are largely blood and to concentrate on the area here dominated by marrow cells adjacent to these more clumped marrow cell areas. Hemopoietic bone marrow has long been the most difficult tissue of the body for histologists to describe, except possibly for the brain. For years, experts made serious misinterpretations about these cells. For example, they erroneously believed that monocytes and lymphocytes were not formed here at all. As I explained earlier, I am not going to distinguish the differentiation pathways for monocytes and lymphocytes in bone marrow. The modern way to identify such cells with certainty is with immunohistochemistry, using antibodies to characteristic cell surface proteins of the individual cell types. Bone marrow is a complex tissue for two reasons. First, it contains entire developmental series of cells from primitive to fully matured forms. And second, 6 types of blood cells listed on this image form simultaneously: erythrocytes, the neutrophils, which are also called heterophils, polymorphonuclear leukocytes or polymorphs, acidophils, which are also called eosinophils, basophils, monocytes, and lymphocytes. So back to the details of this marrow smear. Note the several dark blobs. These are lumps of marrow tissue, too thick to study. And as I mentioned earlier, much of the rest of the smear far away from these lumps is covered mainly with blood, taken inadvertently with the marrow sample. I'm going to zoom in on cells in this area around the blue masses where they're closely packed.